The overall goal of this procedure is to improve the number and diffraction quality of protein crystals obtained from a crystallization screening experiment. To do this, a crystalline or precipitated material is identified from an existing protein crystallization screening experiment established using a target protein of interest. The crystalline or precipitated material is harvested and then used to generate a crystal seed stock. Next, a series of crystallization screens is established containing both the target protein of interest and the crystal seeds. Finally, the seeded crystallization screens are monitored for crystal formation, and any new protein crystals are characterized. Ultimately, the application of this procedure can result in an increase of the number, size, and diffraction quality of protein crystals obtained from crystallization screening experiments.
The main advantages of this technique over existing methods, for example, traditional microseeding or macroseeding, are that it can be performed quickly, is highly scalable, and samples significantly more crystallization space than other seeding methods. Another advantage of this technique is that it does not require the use of high-quality crystalline starting material for seed stock production.
Generally, individuals new to this method will struggle because a significant degree of manual dexterity is required during the crystal harvesting and manipulation steps.
Visual demonstration of this method is critical, as random matrix microseeding deviates significantly from other crystal seeding methods. The subtleties of this method are key to its success, and a number of these are challenging to accurately communicate in text form only.
Begin this procedure with a seeded crystallization tray.